Welcome to El Zoominario. Today, we're very excited to welcome Rene Welch Schwartz. Rene is a computational biologist working at Dr. Irene Ong's lab and supporting Carbone Cancer Center scientists at the University of Wisconsin Madison. Rene is from Mexico City, where he earned his bachelor's in applied mathematics at ITAM and moved to Madison, where he earned a PhD in statistics at UW-Madison. Rene has worked with multiple omics data types, studying disease, cancer, and its association with the immune system. Thank you, Rene. We're very excited to have you here.
Oh, thanks. Thanks for having me. I'm really happy that I got invited to give this talk. I'm going to speak about this project that, well, and certainly it's not completely my project. It's a very large collaborative effort that the lab where I work has been part of. And I'm going to speak about what can we learn from farm kids' immune system when studying house dust microbiome. Basically, what we want to study is the public health concern that is asthma. Like over the years, like people with, well, kids with asthma, allergic rhinitis, or eczema had increased. Right now, allergy prevalence is about like 10 million children. That means that it's 40% of all the children. And the issue with asthma is that it cannot be diagnosed when in babies, right? So when the baby is like very young, you can observe like eczema, which develops into food allergy, hay fever, early life wheezing, and until they are like I don't know, between 6 or 8 years old, they can finally be diagnosed with asthma. So we want to protect that. For this experiment, our hypothesis that microbial exposures in early life are necessary to stimulate the development of a healthy immune system. So that means that, like, if a baby is like exposed to certain bacteria as a baby, that will make the immune system stronger and more tolerant, which in the future, when they grow, that would like protect them from allergies. For this data set, this is, as I said, this is not, I mean, this part of a large cohort effort. This project is an observational bird study cohort. We're trying to identify the specific environmental and immune system factors involving the protective effects of the farm exposure. In particular, we are gathering data, from blood from the babies, stool microbiome, breast milk microbiome, vaginal swabs. In particular, my part of the project is studying house dust, meaning that I'm studying how does the environmental bacteria affects the early Lyme immune system development. Here in the figure, what I'm showing is a county map of Wisconsin, and it's color-coded by the number of dairy farms. In particular, the Herbert cohort has been enrolled in Madison, in Marshall's clinic, where they gather like 150 rural non-farm kids, then 150 non-Amish, we call them traditional agrarian farm kids. And then in Lafarge Clinic, we enroll 50 kids. Obviously, well, of course, even though that this was like the goal of the enrollment, we have even less. Our collaborators, Dr. Chris Ogi and James Kern, are the ones who have collected unique samples from Wisconsin traditional agrarian communities, and the other, these 300 kids that are non-Amish and rural, rural non-farm, they are called as the WIS study. Okay, next. So this is hard experiment. The data was collected basically by leaving these Petri dishes. They were living, well, not Petri dishes all the time, but they were living like the collection devices in the living room. Ideally, that was a place where, it wouldn't be dropped, but it wasn't a place where tons of the dust was going to be, collected. It's not like, next to the broom or next to the vacuum. So this was like in a top shelf or something, or in a bookshelf or whatever. And the idea is that like this device was left there like for, I don't know, 2 weeks or so, around the 2 months time point. Like, well, 2 months from what I'm going to show in these slides, but we had like multiple time points also around 1 year mark, 2 year mark, etc. And whatever dose was collected in this device, that's what is sequenced. The main issue with this is that, how can I say this? Okay, the question is like, how do you measure like all the dust? And hopefully, since you are going to, you know, get sequences of like more than 300 kits, this is supposed to be cheap. So what we use is like 16S ribosomal RNA-seq, where the 16S is a particular region of the bacterial genome that is quite differentiated by different taxonomic ranks, but you don't get to know like the species or the strain. Sometimes you can get like the genera, but that's kind of a lucky case. So what I'm showing here is the image of a sequence file. In particular, this block that I'm highlighting in orange is one sequence. And the part here that are important are going to be two things. One is going to be like the whole sequence. It's the second row here, like CT, C, G, well, etc. And this other sequence below the plus sign, that's going to be the quality scores. And the question here is like, how do we convert this sequence into numbers? Obviously, there are tons of details in the pipeline. We use this tool called Data2, which, you know, without too much detail, what it does is to cluster these sequences into obtaining a list of unique different sequences for all the samples. So this is sort of how our data is going to look like. What I'm showing here is just like the one of our count matrices where the rows are going to correspond to all the babies' houses. Roughly speaking, is going to be one row is one house. I think there are like maybe two households with twins, but like that's negligible. Then the columns correspond to groups or clusters of sequences, and each cell is going to be, right now I'm coloring them as black when there is more than zero number of repeats, and white when it's zero. Now, there are like some rows, for example, here I'm highlighting in orange. Those are the negative controls, and in an ideal world, these rows would be completely white, because like the negative controls, basically what you are sequencing is like, you know, possible contaminants that are in the device that you are using to collect the data. So ideally that would be white, but reality is a bit more noisy. So obviously you can notice that there are like some dust. And finally, the number of sequences are sorted by the total number of repeats. So basically a number like the sum of all the repeats per column. And as you can see, like especially the top, the most repeated sequences, you can see, you know, that are like shaded like more deeply black. But as you go towards the right side of this square of this matrix, it becomes whiter. So yeah. Finally, as part of our pipeline, which I'm not going to mention all the details, each of these sequences can be labeled by a certain bacteria, well, the genera of a bacteria in the most positive case, sometimes species. But that has tons of issues to also solve. So we're going to assume right now that I already did that. So the next question is like, how do we analyze this data? Because we have like over 300 samples, but we have more than 40,000 sequences. So what I'm showing here in this figure is just a summary statistic. In this case, I'm just showing the inverse Simpson. The x-axis is going to be the farm status, which is going to be non-farm with number 2, the rural but non-farm kids. Then number 1 is going to be farm in the middle, and on the leftmost position is TA, which means traditionally agrarian households. On the other hand, the other thing that you can do is check what's called the beta diversity. Here is this is sort of like what was in the statistical field called PCA. This is the principal coordinate components. It's a generalization of that, and the way of analyzing this figure, or the way of interpreting this figure, is that two dots are going to be close to each other if the bacterial composition is similar. So if they share like a lot of the same bacteria, then they're close to each other. If they don't, well, they don't share the same bacteria. And what we can see in both is that there is some sort of progression, right? So for example, here in the inverse Simpson, you can see that like there are like sort of like less bacteria in the non-farm, then there is a bit more on the farm, and then there is significantly more on the traditional agrarian households. And similarly, on the principal coordinates components, you can see that we go from green then to blue, which is the farm kids, well, the farm households, and then the red are a bit more spread out because we have less samples. Okay, so the next thing that we try to do is determine, well, which bacteria are present in each house. And this is the problem. Here what I'm showing is the relative abundance, which means is the frequency of a number of repeated sequences labeled by a different bacteria, different, well, taxonomic rank in this case. And the rows are going to be, again, the traditionally agrarian households here on the top row, then the farm households, and then the rural non-farm households. And from here, we can notice multiple things. One is that there are many bacteria that, at least from a night view, they are not that different in terms of composition. There are ones like, for example, Streptococcus that seems to be like more abundant in non-farm than in farm than in traditional agrarian. Then there is this issue with how you label the sequences, because, for example, it seems like all these Corynebacterium, Corynebacterialis, and Corynebacteriaceae, all three of these taxonomic ranks are in the same lineage. But sometimes the algorithms that you use to sequence can give you these dispersed results. In this case, I believe that I use only one database, but I think that if you gather the results of multiple databases, you can improve these labeling results. And finally, the other issue is going to be this one, that even though like this, I believe there must be like 30-something taxonomic groups that we have here. Still, there are like many more that correspond to more than 40% of the total relative abundance among these families. So the next question is, how can we analyze of this data? Obviously, like classical approaches would say, okay, we can do differential abundance analysis and compare like, like genera by genera, or, order by order, or family by family. But what we decided to use is to predict, to use machine learning approaches to predict farm versus no farm. So what I'm showing here is the curve on the ROC, the x-axis is the 1 minus the specificity, the y-axis is the sensitivity. And these are different models that I use, and different, well, the color are different models. Clear random forest in the blue are the best model like all the time. And the different facets are different ways of summarizing the data. For instance, spray corresponds to these the first 20 components that I'm showing here. Family or genus that correspond to the latest taxonomic rank that I used to gather the data. And log center rank or center log ratio are different transformations of the data. Regardless, it seems that in all of the cases, it seems that in all of the cases, random forests are the best model. So if we analyze and search for the top 25 bacteria that are more important when predicting, we can see that there is a very clear separation, for example, in Moraxella or DHC or many of them, between farm households and non-farm households. And each panel at the end measures how those given taxa is affecting the predictions layered by a model. Here, I forgot to mention, here I'm just plotting the partial dependence curves. It's a viral importance metric. And the most, what we use to measure the viral importance is basically the range, the vertical range between the top value and the lowest value. But this is not all. At the end, this is the most important part of this is this result. What we did in this analysis is we fitted the logistic regression models where the response is going to be 0 or 1. And that means the eczema has been reported at like a certain time point or before by their families. This is the columns of these panels is how we used, how we transform the covariates to predict the probabilities of the households to predict the probability of being farm on the non-farm households, and then we aggregate the rows are how we aggregated the taxonomic ranks. And what we're plotting on the x-axis is the odds ratio of this prediction model. And the interesting part are the error bars in blue, because that means that the odds ratio is below 1, which in turn means that there is a negative correlation between getting diagnosed with eczema in the future when the environment of these non-farm households is more farm-like, which makes us think that at the end, that may mean that there must be like some protective effect in the non-farm households when they look more like farm. So that must be, well, that's all. I would like to thank my collaborators. Irene Ong, she's the PI of my lab, and the Lab and SolisLemus-lab, which are our main collaborators. That's it.